4 billion years ago, Earth was already a world transformed. The violent bombardments that once scarred the surface had slowed. Oceans wrapped the planet in shimmering blue. And inside those oceans, after hundreds of millions of years of molecular trial and error, protocells had formed, fragile droplets of chemistry capable of growth, division, and crude forms of evolution. But protocells were only the beginning. They were unstable, simplistic, vulnerable to heat, salinity, UV radiation, and the chaos of early Earth. Their membranes were leaky, their internal chemistry easily disrupted, and their genetic molecules too primitive to store large amounts of information. They were a transitional form, an early step towards something far more organized, resilient, and powerful. Life was close, but not here yet. For the first true cells to emerge, an extraordinary transformation had to occur, a transformation that required new molecules, new structures, and new rules of organization. The ancient oceans now became a vast laboratory filled with protocells drifting aimlessly, colliding, splitting, dissolving, recombining, and undergoing millions of chemical experiments every second. Most vanished without a trace. A few survived long enough to accumulate small advantages. And over unimaginable spans of time, those advantages began to stack. Primitive genetic strands, RNA-like molecules capable of copying themselves, became more stable inside protective membranes. Some protocells absorbed mineral particles that acted like catalysts, speeding chemical reactions. Others captured molecules that improved their ability to produce energy-rich compounds. The earliest forms of metabolism, still chaotic, still fragile, began to take root. But evolution was about to push these systems to the next level. Some protocells discovered ways to repair their own damaged strands. Others developed networks of reactions that allowed them to extract energy more efficiently from their surroundings. These subtle improvements reshaped their internal chemistry, making them less dependent on random environmental conditions. Slowly, a stable internal environment began to emerge, a space inside the membrane where chemical reactions could happen reliably, shielded from the chaos outside. This was the birth of protocytoplasm, the earliest precursor of the internal environment of living cells. At the same time, another revolution occurred. Instead of the simple leaky membranes made of fatty acids, some protocells began to incorporate more complex molecules. Lipids with two tails instead of one, molecules far better at forming durable, self-sealing layers. This created membranes that were more stable, less prone to falling apart, and capable of maintaining a strong internal difference between the inside and outside world. This was not merely a structural improvement. It was a major evolutionary event. Protocells with these improved membranes could survive hotter environments, saltier water, stronger currents, and more ultraviolet radiation. They could hold on to important molecules instead of leaking them into the ocean. They could start to act like enclosed systems, microworlds cut off from the rest of the ocean. The first true cell was now within reach, but the biggest breakthrough of all came in the form of a molecular partnership. RNA molecules were good at storing information and guiding reactions, but they were unstable and limited in what they could do. Proteins, on the other hand, were extraordinary tools, capable of folding into complex shapes, forming enzymes, speeding reactions by orders of magnitude. But proteins could not copy themselves or store information. The solution to this problem was one of the greatest innovations in the history of Earth, a division of labor between two molecular worlds. RNA kept the instructions, while protein enzymes began to handle the heavy chemistry. This was the earliest form of the genetic code, not yet the same code used by all life today, but an ancestral version of it. As protocells evolved this partnership, their internal reactions became faster, more coordinated, and more flexible. RNA strands that once struggled to copy themselves now had enzymatic helpers. Proteins that once formed by chance now formed through guided sequences. For the first time, information became the driver of chemistry. Natural selection began to favor protocells that stored clearer instructions, produced better catalysts, and maintained more stable membranes. And then, somewhere in Earth's ancient oceans, perhaps near a hydrothermal vent, perhaps in a tidal pool rich with mineral scaffolds, One protocell crossed the final threshold. It assembled a working genetic code, stable energy pathways, durable membranes, and a system capable of copying both itself and the instructions that made it. It was no longer a protocell, it was a true cell. This cell was not alone for long. It divided, it drifted, it found chemical resources. It changed, adapted, expanded, and over time it became the last universal common ancestor, the organism from which every living thing on Earth would eventually descend. LUCA was not the first living cell, but it was the only survivor of its era, whose lineage continues today. Countless other early cells evolved, thrived for a while, and vanished without descendants. But LUCA's lineage endured, spreading through the oceans, evolving countless metabolic strategies, and building the foundations of every biological system that would follow. This episode begins with a world still ruled by chemistry, but ends with biology taking command. The rise of the first true cells is one of the greatest transitions in the history of Earth, a moment when the planet itself learned how to copy information, harness energy, and engineer complexity. And this transition set the stage for everything else: microbes, photosynthesis, ecosystems, multicellular organisms, animals, plants, and eventually the human mind that now looks back across 4 billion years to witness the moment where life truly began. The story of LUCA is the story of every living thing. Life's earliest chapter had now shifted from fragile protocells to primitive true cells. But the world these first cells entered was anything but gentle. Early Earth was still a harsh planet, hotter, more volcanic, more chaotic than anything seen today. Yet inside this unforgiving environment, these new cells began to carve out microhabitats where stability could exist. Around hydrothermal vents, in pooled waters rich with minerals, and beneath protective layers of sediment, cells found places where energy sources were abundant and where dangerous ultraviolet radiation could not easily reach. These environments became the training grounds for the first generations of true living systems. Unlike protocells, the new cells had internal structures that allowed them to regulate their chemistry more systematically. Their membranes were far better at separating the inside from the outside, allowing the interior to maintain a balanced environment even when the ocean around them shifted dramatically. Inside these first cells, chains of chemical reactions, primitive but structured, began to operate continuously. Instead of waiting for random environmental changes to drive reactions, the cell itself now guided and controlled its own internal chemistry. This was independence. This was the beginning of biological autonomy. One of the most important innovations these early cells developed was a reliable way to extract energy. Protocells depended heavily on the environment for energy-rich molecules, but true cells evolved their own internal systems to create or capture energy. Some cells began using geothermal chemicals, reacting gases like hydrogen and carbon dioxide to produce energy-rich molecules. Others exploited gradients across their membranes, differences in acidity or electrical charge, to power molecular machinery that produced energy internally. This was the ancient origin of the processes that all modern life still relies on today. Proton gradients, electron transfer chains, and metabolic pathways that allowed cells to power themselves in a stable, predictable way. As the first cells refined these early metabolic strategies, evolution began to accelerate. Cells with faster energy production survived better. Cells with more stable membranes reproduced more reliably. Cells with improved genetic copying passed on more accurate information. Slowly, the early genetic systems became more complex, more reliable, and more capable of building proteins with precise shapes and functions. These proteins, in turn, made the cells even more efficient, creating a positive feedback cycle that pushed early life into increasing complexity. But despite these advances, life was still balancing on the edge of stability. The environment could shift without warning. Temperatures could spike, minerals could fluctuate, toxins could sweep through hydrothermal fields. Entire early lineages of cells likely rose and fell. Within mere thousands of years, barely a blink on geological timescales. Evolution, however, thrives on instability. Each collapse opened ecological space for something new to emerge. Each environmental shift forced cells to adapt or perish. Over time, only the most resilient strategies remained. During this period, early cells also began forming more stable communities. Rather than existing as isolated individuals, they grouped near mineral structures, film-like surfaces, and porous rocks that offered protection and resources. In these microhabitats, cells exchanged chemicals, shared byproducts, and inadvertently created cooperative networks. Some cells released molecules that acted as nutrients for others. Some stabilized the environment around them. Some produced waste gases that became valuable fuel for neighboring cells. These accidental forms of cooperation laid the groundwork for future ecological systems, a preview of the interconnected web of life that would one day fill the oceans, land, and air. Another major evolutionary milestone occurred when early cells refined their use of RNA and proteins. Over time, DNA began to appear as a more stable storage medium for genetic information. DNA was less reactive than RNA and could store much longer sequences without degrading. Cells that adopted DNA gained a huge advantage. They could encode more instructions, build more proteins, and develop more complex metabolic networks. With DNA came the earliest cellular genomes, tiny by modern standards, but revolutionary in their capacity to store and protect information. As DNA-based systems became more common, the ancient microbial world began to diversify. Different cells adopted different metabolic strategies, different habitats, and different membrane chemistries. Some cells thrived near heat. Others preferred cooler waters. Some relied on hydrogen for energy. Others used sulfur, iron, or carbon compounds. This was evolution's branching point, where the tree of life first began to split into separate domains, although LUCA itself still lay ahead, representing the last common point before those great divergences. But before LUCA's lineage rose to dominance, countless early life forms coexisted in the ocean, some stable, some transient, all contributing to the chemical landscape that shaped the next evolutionary steps. The oceans were thick with microscopic life, though invisible to the naked eye. Entire worlds existed in droplets of water, on mineral grains, within cracks of rock. The surface of the planet was still barren, but underwater, life was beginning its quiet conquest. This era was also marked by an explosion of molecular experimentation. Cells tinkered with new proteins, new membrane structures, and new ways of processing carbon. Some early lineages attempted photosynthesis-like processes, though true oxygen-producing photosynthesis would not appear for hundreds of millions of years. Others evolved primitive forms of fermentation. Some refined their energy systems so much that they became capable of surviving in environments nearly devoid of external fuel. Evolution was innovating constantly, testing every possible route, keeping only the versions that worked best. As the earliest cellular ecosystems expanded, competition emerged for the first time. Cells that could grow faster outcompeted slower ones. Cells that could repair damage more quickly survived environmental shocks. Cells with more robust genomes outlasted those with sloppy copying mechanisms. But this competition also drove innovation. Each metabolic improvement in one lineage pushed others to adapt or fade. Life was no longer just reacting to the environment. Life was now reacting to itself. Amid this chaotic, competitive, experimental world, a single lineage began to rise, one that would eventually become LUCA. We do not know what this organism looked like. No fossil remains exist from this era. But by comparing the genetic machinery shared by all modern life, we can reconstruct parts of its biology. LUCA used DNA to store genetic information. It used RNA to transfer instructions. It used proteins shaped by a universal genetic code. It used proton gradients across its membrane to power its internal reactions. It lived in an environment rich with hydrogen and carbon dioxide, likely near alkaline hydrothermal vents where conditions were stable enough for long-term survival, but dynamic enough to fuel metabolism. LUCA was not the beginning of life. but it was the beginning of the lineage that would lead to everything alive today. As its contemporaries vanished, LUCA's descendants spread through the oceans, adapting to new environments, diversifying into the ancestors of Bacteria and Archaea, and laying the next layer in the foundation of Earth's living systems. Life had crossed the threshold. The age of cells had truly begun. As early cells continued to multiply and spread across the oceans, evolution shaped them with relentless precision. Every mutation, every copying error, every slight change in protein shape or membrane structure offered a new experiment. Most of these experiments failed, but every so often, one succeeded. And when it did, the descendants of that cell carried the improvement forward, reshaping the biological landscape for millions of years to come. This period was not a sudden burst of innovation, but a steady, powerful accumulation of improvements that gradually separated the earliest advanced cells from the simpler protocells that came before them. One of the most important transformations involved the control of chemical gradients across the membrane. Early cells began using proteins embedded in their membranes to shuttle ions in and out, creating electrical and chemical differences that could be used as energy. This internal battery-like system allowed cells to maintain a constant flow of energy regardless of the conditions outside. Hydrogen ions, in particular, became central to this process. Cells that mastered the ability to pump protons across their membranes could harness that gradient to power the molecular machinery inside. This ancient mechanism, refined over billions of years, still exists in modern organisms as the backbone of cellular respiration. But the development of controlled energy systems was only one piece of the puzzle. Another major innovation was the evolution of ribosomes, complex molecular machines capable of reading RNA instructions and building proteins with remarkable accuracy. Ribosomes likely developed gradually, starting as small clusters of RNA molecules that stabilized each other and assisted in simple reactions. Over time, they incorporated proteins, evolved more precise binding sites, and became capable of producing longer and more complex proteins. Once ribosomes reached a certain level of sophistication, they allowed early cells to create enzymes far beyond what simple, random chemistry could produce. The emergence of ribosomes was one of the most important steps in the history of life because it gave cells access to an entire universe of possible protein shapes and functions. With these improved tools, early cells began constructing internal networks of reactions that worked together to keep the cell alive. Instead of relying on a single pathway for building molecules, they evolved branching pathways where one reaction fed into the next. These metabolic cycles became more efficient over time, allowing cells to reuse resources, minimize waste, and adapt quickly to changes in the environment. For example, if a particular nutrient became scarce, some cells evolved pathways to produce that nutrient internally. If toxins accumulated, other cells developed proteins capable of breaking them down. Life was learning how to engineer its own environment from the inside. At the same time, DNA underwent a transformation of its own. Early genomes were small and fragile, but as cells developed better repair mechanisms, they could expand their genetic libraries. Repair enzymes that recognized and corrected mutations appeared, along with mechanisms that detected mismatched base pairs or damaged chemical structures. These innovations reduced the number of harmful mutations and allowed beneficial mutations to accumulate more predictably. Over countless cycles of copying, repairing, and refining, the accuracy of genetic replication increased dramatically. This higher fidelity unlocked the ability to store larger and more complex sets of instructions, enabling the evolution of more advanced proteins and metabolic systems. As genomes expanded, cells began to specialize in different ways of obtaining energy. Some cells focused on chemiosynthesis, using hydrogen, methane, or sulfur compounds. Others adapted to slightly cooler environments and evolved metabolic pathways that relied on carbon compounds dissolved in the ocean. A few even began experimenting with early forms of anaerobic respiration, extracting energy from interactions that produced minimal waste. These metabolic differences created the first ecological niches, microbial territories defined not by land or geography, but by chemical availability and environmental conditions. Because early Earth's oceans were rich in minerals, metals, and dissolved gases, early cells often incorporated these materials directly into their enzymes. Iron, nickel, cobalt, and sulfur all played crucial roles in early metabolism. Some enzymes formed metal clusters at their cores, using these reactive elements to speed up chemical transformations. This relationship between metals and enzymes became so essential that traces of it can still be found in the biochemistry of modern organisms. Even today, your cells use iron-based enzymes for energy production, a direct inheritance from the chemistry of Earth's earliest life. As the oceans became populated by these early cells, interactions between them grew more complex. Some cells engaged in competition, releasing molecules that hindered the growth of others. Some developed the earliest forms of chemical communication, sensing changes in their environment caused by neighboring cells. Others produced sticky molecules that allowed them to attach to mineral surfaces, forming biofilm-like communities that offered stability and shared protection. These early microbial ecosystems laid the groundwork for the vast and interconnected biosphere that Earth would one day support. Eventually, through the sprawling network of competition, cooperation, and environmental pressures, one lineage began to stand apart from the rest. Its genetic code grew more refined, its enzymes became more efficient, its membranes became more stable and better at maintaining internal chemical gradients. Its ribosomes grew more sophisticated, allowing it to produce proteins with exceptional reliability. This lineage was not yet LUCA, but it was heading directly toward that evolutionary milestone. The cells of this lineage also developed highly efficient methods for copying DNA. They evolved helicases to unwind the double helix, polymerases to build new strands, and proofreading mechanisms to check for errors. The entire copying process became more coordinated, ensuring that each generation inherited a complete and largely accurate set of genetic instructions. This breakthrough massively accelerated evolution because it allowed beneficial changes to spread through populations while reducing the chaos caused by harmful mutations. By this point in Earth's history, nearly every major component of modern cellular life had emerged in some form. The genetic code, while still incomplete, was almost universal. Ribosomes were nearly fully formed. Membranes were stable and selective. Metabolism was sophisticated enough to support diverse lifestyles, and energy production relied on proton gradients, just as it does today. What remained was the final consolidation of these features into a single organism whose descendants would outlast all others. That organism would become LUCA. As early cells continued refining their internal chemistry and expanding their genetic capabilities, the oceans around them became increasingly influenced by their presence. Life, even in its simplest microbial form, was already beginning to modify the planet. The earliest cells released metabolic byproducts that accumulated in the water around them, subtly reshaping local chemical gradients and altering the availability of key nutrients. These changes created entirely new microenvironments, zones where certain chemical resources became concentrated, where energy sources fluctuated, and where different types of microorganisms could thrive or falter depending on their genetic tools. This shifting landscape drove further diversification, setting the stage for the next major evolutionary developments. One of the most profound shifts during this era was the emergence of specialized transport systems. Early membranes were becoming more selective as they incorporated complex two-tailed lipids and protein channels, but the real leap occurred when cells evolved transport proteins that could actively pull nutrients inside or push waste products out. These transporters were essential for maintaining homeostasis in environments that fluctuated rapidly. A cell that could selectively import key molecules like hydrogen, carbon dioxide, nitrogen compounds, or metallic ions gained a tremendous advantage over those still relying on passive diffusion. This ability essentially allowed cells to take command over their internal environment, a level of control protocells could never achieve. Alongside these transport systems, cells began developing far more intricate metabolic pathways. Instead of relying on a handful of chemical reactions, advanced early cells started to build multi-step cycles that reused chemical intermediates and produced consistent streams of usable energy. The earliest versions of pathways that resemble modern glycolysis, carbon fixation, and amino acid synthesis likely emerged during this time, not identical to the versions used by modern organisms, but recognizable in their logic and structure. Each pathway added a new layer of efficiency, allowing cells to survive in environments where resources were scarce or unpredictable. Another critical innovation involved the stabilization of ribozymes and their transition into mixed RNA-protein complexes. RNA still played a key role in catalyzing reactions, especially during protein synthesis, but proteins began taking over as the primary workhorses of the cell. The chemical diversity offered by 20 amino acids allowed proteins to fold into shapes RNA could never achieve, giving rise to enzymes that were faster, more precise, and far more adaptable. This shift marked a turning point in the balance of molecular power. RNA became the carrier of information, while proteins became the executors of that information. This arrangement was so effective that it has remained the foundation of life ever since. But even with these advances, early cells faced relentless threats. UV radiation from the young sun constantly bombarded the ocean surface, damaging DNA and breaking apart fragile molecules. Thermal vents produced eruptions of scorching water that could wipe out entire populations. Volcanic activity introduced toxins into the oceans, creating chemical barriers that only the most resilient cells could cross. Yet, with every threat came an opportunity for adaptation. Cells that evolved better DNA repair systems gained a survival advantage. Cells that found safer microenvironments or developed protective molecules could endure conditions that destroyed their competitors. Evolution was sculpting the early microbial world through an unending series of environmental trials. Eventually, the lineages leading toward LUCA began to develop some of the core housekeeping mechanisms that define all modern life. They refined their genetic code, making the mapping between codons and amino acids more reliable. They improved their replication machinery, reducing the frequency of harmful mutations. They perfected the structure of their ribosomes, enabling them to build complex proteins with extraordinary speed and accuracy. And they developed enzymes capable of regulating metabolism, repairing cellular structures, and managing stress responses. These systems did not appear all at once. They accumulated slowly, forming a molecular architecture that grew more resilient with each generation. A particularly important development was the evolution of membrane-associated proteins responsible for maintaining proton gradients. These proteins formed the earliest versions of the molecular pumps and rotors that would later become the ATP synthase complexes found in all living cells. These enzymes allowed cells to convert energy stored in proton gradients directly into chemical energy in the form of ATP, a molecule that would become the universal energy currency of life. Although LUCA itself may not have used ATP synthesis exactly as modern organisms do, it almost certainly relied on a similar system based on proton gradients and energy-carrying molecules that performed comparable roles. At this stage, cellular life was becoming increasingly robust and microbial communities were spreading across the ocean floor. Life had not yet reached the surface waters in significant numbers. The radiation was still too intense. But sheltered environments like hydrothermal fields provided stability, chemical resources, and protection. These environments became evolutionary hotspots where countless lineages rose and fell. Some of these lineages developed traits that were incredibly advanced for their time, but most eventually disappeared, leaving no descendants. Only one lineage persisted, carrying the core features that would define all future life. This lineage was tightening its grip on survival, building a molecular toolkit that could withstand the challenges of early Earth with remarkable efficiency. Yet despite its growing resilience, this lineage still existed within a world of genetic chaos. Horizontal gene transfer, where cells exchanged genetic material without reproduction, occurred frequently. Genes passed from cell to cell, crossing boundaries and spreading innovations through entire communities. This exchange accelerated evolution by making successful adaptations available to neighboring organisms. It also created an environment where lineages blurred together, sharing and reshaping their molecular machinery. LUCA was not a single isolated organism emerging from a tidy evolutionary tree. It was the product of a dynamic network of gene sharing, mutation, competition, and cooperation. By the time LUCA's lineage finally emerged as the dominant survivor of this era, it carried with it a collection of systems that had endured millions of trials. It had a stable DNA genome, a reliable genetic code, a fully functional ribosomal machinery, and a membrane capable of maintaining strong chemical gradients. It had metabolic pathways that extracted energy from hydrogen, carbon dioxide, and mineral reactions. It lived in a world already shaped by its ancestors and contemporaries, an ocean filled with chemical diversity and microbial experimentation. LUCA was not the first cell, but it was the first cell whose descendants would populate every branch of the tree of life. By the time LUCA, the last universal common ancestor, stood at the threshold of evolutionary history, the microbial world had already become a complex network of chemical ecosystems. The oceans were no longer just environments of raw geology and simple chemistry. They had been transformed by billions of years of microscopic activity. Compounds that had once entered the oceans solely through volcanic eruptions or meteorite impacts were now being cycled, processed, and repurposed by living cells. Life had become a geological force. LUCA did not arise in isolation. It emerged from a vibrant, competitive, highly dynamic microbial world where horizontal gene transfer blurred the boundaries of lineage and heritage. Genes could be inherited from ancestors or acquired from neighbors, meaning evolution in this stage of Earth's history was not a tree but a tangled web. LUCA was not an organism with traits never seen before. It was a survivor whose molecular machinery had been tested across countless environmental challenges. The features it carried were those that proved universally resilient. One of the most defining aspects of LUCA was its sophisticated genetic system. It possessed a DNA-based genome stabilized by a full suite of enzymes responsible for replication, repair, and transcription. These enzymes were more precise than those of the earliest cells, reducing the frequency of catastrophic mutations. LUCA used a universal genetic code nearly identical to the one used by every organism alive today, which allowed it to translate sequences of nucleotides into sequences of amino acids. This code was not perfect, but it was robust, and its consistency allowed proteins to evolve more rapidly without destabilizing the entire system. LUCA's ribosomes, while primitive compared to modern versions, were fully capable of building complex proteins with a high degree of accuracy. These ribosomes represented an evolutionary victory. They were efficient enough to support fast growth, yet stable enough to maintain fidelity across generations. The foundational architecture of all modern ribosomes was already present: large and small subunits, rRNA components, and essential structural proteins. The machinery of translation, a process older than LUCA itself, had reached a level of refinement that would remain virtually unchanged for over 4 billion years. Membrane biology also reached a critical turning point. LUCA's membranes were fully stable, composed of chemically robust lipids that allowed the cell to maintain strong proton gradients. These gradients provided the energy needed to power the earliest ATP-producing systems. Although LUCA's exact energy metabolism is still debated, it almost certainly relied on redox reactions involving hydrogen, carbon dioxide, and metal ions, chemical currencies abundant around hydrothermal vents. Its membrane proteins were capable of shuttling protons, managing ion concentrations, and regulating the flow of metabolic intermediates. This level of membrane control gave LUCA the ability to thrive in environments where chemical gradients shifted rapidly. LUCA also carried enzymes for carbon fixation, likely through a primitive version of the Wood-Lungdahl pathway, a metabolic route that allows organisms to convert carbon dioxide into organic molecules using hydrogen as an energy source. This was not photosynthesis. Sunlight played no role here. Instead, LUCA exploited the raw chemistry of the deep ocean, tapping into energy sources that were stable, abundant, and independent of surface conditions. This reliance on geological energy, rather than solar power, made LUCA resilient to catastrophic events happening at the surface, including asteroid impacts and fluctuations in the early atmosphere. One of LUCA's most remarkable abilities was its capacity to repair itself. UV radiation, volcanic chemicals, and spontaneous mutations constantly threatened the integrity of its genome. LUCA's DNA repair enzymes, primitive but effective, allowed it to detect and correct many of these faults before they became lethal. The system did not prevent evolution, it moderated it, ensuring that change occurred at a sustainable pace rather than through destructive chaos. These repair mechanisms provided the stability necessary for long-term survival and diversification. LUCA also had a complex internal chemistry. It used cofactors, small molecules that assisted enzymes in catalyzing reactions. Many of these cofactors, such as iron-sulfur clusters, NAD-like molecules, and early versions of coenzyme A, are still used by every living organism. Their presence in LUCA indicates that these molecules were evolutionary successes long before LUCA itself existed. The biochemical toolkit that LUCA inherited was a refined collection of the most reliable molecular solutions evolution had produced up to that point. Yet, despite these advances, LUCA was not the pinnacle of early evolution. It was simply the lineage that managed to endure. Many lineages of early cells were just as sophisticated, some perhaps even more so in specific traits. But they eventually died out as environments shifted, resources dwindled, or competition intensified. LUCA's survival was partly due to luck, and partly due to a balanced combination of traits: stable genetics, reliable metabolism, resilient membranes, and efficient protein synthesis. LUCA was not a mother of all life in the poetic sense. It did not give birth to all future organisms through direct reproduction. Instead, LUCA represented the last point at which all life shared a common, interconnected genetic pool. After LUCA, the evolutionary web began to branch irreversibly. Populations became isolated, mutations accumulated independently, and genetic drift gradually separated lineages into distinct groups. From these splintering lineages, the first two great domains of life, Bacteria and Archaea, began to take shape. LUCA lived in a world of geothermal energy, mineral-rich waters, and constant chemical flux. Its life was simple, but remarkably stable. It had no nucleus, no internal compartments, and no specialized structures beyond its membrane and molecular machinery. Yet within this modest framework lay the potential for everything that would follow: photosynthesis, respiration, multicellularity, complex ecosystems, and eventually the rise of animals, plants, and humanity. LUCA was the hinge of biological history, the point where the story of life unified before branching into every form that has ever existed. LUCA did not vanish in a single moment, nor did the split between its descendants happen cleanly or suddenly. Instead, the branching of life into Bacteria and Archaea unfolded gradually, through countless generations of cells exchanging genes, adapting to different environments, and slowly drifting apart until they no longer resembled the same organism. Part 6 is where LUCA's unified world begins to fracture into two fundamentally different biological empires, each with its own strategies for survival, its own molecular identity, and its own evolutionary destiny. At first, LUCA's descendants still lived in overlapping habitats. They shared hydrothermal vent systems, exchanged genetic material, and competed for the same chemical resources. But subtle differences in their internal machinery began to accumulate. These differences, though invisible to the naked eye, altered how each lineage interacted with the environment. Small changes in membrane chemistry, enzyme structures, and metabolic pathways created divergent strategies for handling stress, processing nutrients, and maintaining energy balance. One of the earliest divergences occurred at the level of membrane structure. LUCA's descendants began experimenting, through mutation, selection, and horizontal gene transfer, with new lipid chemistries. Some lineages evolved membranes built from ether-linked lipids. a structure far more stable at extremely high temperatures. These lineages gradually became the ancestors of the Archaea. Their membranes were rigid, chemically resilient, and perfectly suited for surviving scalding hydrothermal fluids, acidic environments, and rapid temperature fluctuations. This allowed early archaeal cells to push deeper into extreme settings where other microbes struggled to survive. Meanwhile, other LUCA lineages developed a different solution: ester-linked lipids, which created more flexible membranes. Though less heat-resistant, these membranes worked beautifully in cooler, more chemically diverse environments. These lineages drifted toward what would ultimately become the bacteria. They were not extreme environment specialists, they were versatility specialists, and that versatility allowed them to spread out, colonize new habitats, and exploit resources that archaeal ancestors ignored. Another divergence unfolded in their genetic and metabolic machinery. Archaeal ribosomes, transcription systems, and DNA handling enzymes evolved increasingly unique structures, more similar to those found in modern eukaryotes than to bacteria. Their enzymes became optimized for extreme conditions, allowing them to maintain stable function in boiling water or super salty pools. Bacterial ancestors, by contrast, refined their biochemical systems for speed and efficiency. Their enzymes were fast, flexible, and adaptive to a wide range of conditions, giving them a competitive advantage wherever rapid growth could outpace environmental hazards. Even their metabolisms diverged. Archaeal ancestors specialized in utilizing unusual fuels, methane, sulfur compounds, highly reduced minerals, and chemical gradients found at the ragged edge of habitability. Bacterial ancestors leaned into pathways that allowed rapid biomass production, fermentation, photosynthesis-like innovations, and early versions of respiration. As environments shifted across the planet, each lineage found its own ecological niche. This divergence also reshaped the genetic landscape. Gene exchange, the chaotic genetic marketplace that had defined life before LUCA, slowed. It didn't stop entirely, but as membranes, enzymes, and cellular machinery grew more distinct, cross-lineage compatibility dropped. Genes that functioned flawlessly in one lineage often failed in the other because the surrounding cellular context had changed too much. This incompatibility created a natural barrier between the emerging domains. By the time this barrier fully solidified, the two lineages had diverged beyond return. What makes this transition so extraordinary is that it set the stage for everything life would later achieve. The bacteria, with their flexible membranes, fast metabolisms, and high adaptability, became the great expanders. They spread into every ecosystem, from deep-sea sediments to shallow tidal pools to the surface of rocks exposed to sunlight. They would eventually invent oxygenic photosynthesis, fundamentally reshaping the planet. The Archaea, with their stability and resilience, carved out domains that would otherwise remain uninhabitable. They became the masters of extremes, boiling springs, methane-rich muds, the deepest and darkest crustal reservoirs. Some archaeal lineages even laid the molecular groundwork for the future emergence of complex cells, the ancestors of all animals, plants, fungi, and protists. And all of this began with subtle, almost undetectable differences, but accumulated gradually over immense spans of time. By the end of this transitional era, Earth no longer hosted a single unified microbial lineage. It hosted two massive branches of life, two fundamentally different biological blueprints, two worlds that would coexist, compete, and collaborate through the next 4 billion years of evolution. LUCA itself faded into history, not through extinction, but transformation. Its descendants had become something new. The unity that once defined life had ended. The diversity that would one day give rise to everything, from trilobites to dinosaurs to humans, had begun. Once the early descendants of LUCA diverged into the first true Bacteria and Archaea, the planet itself began to change in response. These lineages were not simply passive organisms surviving in isolation. They became geological forces, reshaping oceans, rocks, minerals, and even the chemistry of the atmosphere. This part of the story is where life stops being a passenger on Earth and becomes one of its primary architects. As bacterial ancestors spread into cooler, shallower, more chemically diverse waters, they began pioneering new kinds of metabolism. Some experimented with primitive versions of photosynthesis. This early photosynthesis did not yet produce oxygen, but it allowed these microbes to harness sunlight. Every innovation gave bacteria another advantage. They could grow faster, colonize faster, and harvest energy from expanding environments. The world's surface waters slowly shifted from purely chemical-driven ecosystems to ones where sunlight played a growing role. Archaea, meanwhile, continued to thrive in extreme environments: superheated vents, acidic pools, methane-rich sediments deep beneath the seafloor. Their metabolisms began to stabilize global chemical cycles. Methanogenic archaea released methane into the oceans and atmosphere, influencing the greenhouse effect and keeping the young Earth warm despite its faint early sun. Without these archaeal ancestors, the planet might have frozen long before complex life had a chance to evolve. As the two domains expanded into their respective niches, competition increased. Bacterial lineages with faster replication and higher genetic turnover often outcompeted other microbes in nutrient-rich environments. But archaea excelled where survival depended on endurance rather than speed. The planet became a patchwork of microbial ecosystems, each dominated by whichever lineage was best adapted to the specific conditions. Genetic separation between the two domains further solidified. Their ribosomes, membranes, and enzymes continued evolving along separate trajectories. Bacteria refined their cell walls and structures, creating biochemical solutions that would one day enable them to build biofilms, mats, and colonies that trapped sediments and reshaped entire landscapes. Archaea developed enzyme systems capable of withstanding pressures and temperatures that would shred bacterial proteins. The split also created evolutionary playgrounds. Once isolated from each other's gene pools, each lineage explored its own evolutionary space. For bacteria, this meant absorbing foreign genes, swapping DNA rapidly, and experimenting with new biochemical innovations. For archaea, this meant refining stability, protection, and energy extraction from exotic sources like hydrogen, ammonia, and deeply buried minerals. Slowly, through millions of years, Earth transformed into a planet divided into two great microbial empires. And though the split between bacteria and archaea may seem like a small technical detail, just a change in membrane chemistry, just a shift in enzyme structures, it was one of the most important events in the history of life. Because from this divergence came everything else. The bacteria would someday produce the oxygen that shocks the planet and triggers the great oxidation event. The archaea would someday give rise to the lineage that becomes the first complex cells, the ancestors of all higher life. Every plant, every animal, every fungus, every human would ultimately trace their lineage back to an archaeal ancestor that emerged long after this divergence. This split, the first major branching of the tree of life, marked the beginning of biological diversity. LUCA's world had been unified. The world after LUCA was forever divided, shaped by two evolutionary paths that would never merge again, yet would collectively define the next 4 billion years of Earth's story. And with this, the chapter ends. LUCA's descendants have parted ways. Bacteria and archaea now rule the young Earth. And the stage is set for the next great revolution in the history of life.